Now, this session is not just going to be about cell transplantation specifically, but rather about some of these complex advanced surgical approaches that are being taken towards Parkinson's. And we'll talk about deep brain stimulation. We'll talk about cell transplantation. We'll talk a bit about gene therapy as well, maybe. So what I will do is I will kick off a couple of first questions. And I'll start with Roger, just following on from the previous session. We'll stay with the theme of cell transplantation, having just said we're not going to talk about cell transplantation specifically. But Roger, where do you, if the Blue Rock Therapeutics phase 3 trial is successful, and the regulators say this is fantastic result, we will approve this for clinical use. Where do you see, how do you see the rollout of this as a therapy for Parkinson's?
So if all of those ifs are true, so it's a positive phase 3 study showing that it has a significant benefit, then that will obviously then get a license for people with moderately advanced disease who are looking towards some therapeutic intervention. So it'll be indicated for Parkinson's and it'll be indicated for that stage. That would be my understanding. And then I imagine they would try and move it into the European market. Then I think the question is, well, where would it go from that? And I suspect if you look at something like deep brain stimulation, this would be a similar sort of pattern, if you like. So you would say, well, it's very effective. It would obviously have to compete with the other therapies that are out there. And one thing we haven't mentioned is the cost. So obviously the cost of whatever this therapy then becomes, whatever it is, will have to be factored into whether people can afford it, and especially in the UK, whether we could afford that. But ultimately, I think if it really worked, then there is an argument to be said, and probably more so than for deep brain stimulation, that you simply move it earlier into the disease course. So, my ultimate vision would be that if this really works, it would be a first-line treatment for people, because if it really works, then in the early stages, you don't need very much. And you could probably say, I'll put up with it for 2 or 3 years whilst this transplant works. Once the transplant works, I don't need to take any drugs for the dopamine system because the transplant's doing it. If I don't take drugs, I don't get complications. If I don't get complications, I don't need the treatment for the complications. So in essence, if it works, in theory, it would remove the need for any of the therapies we currently have for Parkinson's. That's the vision.
If I may add to that.
Please.
That was one of the biggest decision makers in terms of the thought process to catch the disease early, in that it could keep people in work without having to give up work if you catch it early enough, and then give them a much fuller life for longer, rather than waiting till you've got to the kind of lowest threshold that you're willing to put up with, and then kind of get an intervention at that point. So I think that was a really important difference between some of that and some of the other studies that STEM-PD is trying to approach.
And just thinking about this slightly more broader in terms of single-payer healthcare system, for example, in Copenhagen, in Denmark, Agnete, how do we? Roger's partly answered my question, but not fully. How do we foresee the rollout of this as a therapy for a healthcare system?
Yeah, I mean, I think you're asking a very relevant question, because it's not easy, right? You can't go to the pharmacy and pull it off the shelves and go home and administer it to yourself. It requires a huge setup, right? You need neurosurgeons, nurses, hospitalization for several days. And that is something, there is no quick fix, right? We don't have a way to get around that. You can't just inject these cells into the bloodstream. I do think it can still be cheaper, actually, than many other therapies, because it's a one-time treatment. That is at least the idea. These cells are supposed to stay in the brain and work for the lifetime of the patient. So I think when you add that up to 10 or 15 years of other therapies, I do think it can be competitive. But in the end, we are not the ones setting the price for a therapy like this. That will be the pharmaceutical companies. And that is why I am actually happy to see that there is more than 1 or 2 trials. There's a good amount of competition in this field. And I think that's healthy. And I think it's good for the patients because competition is the only thing that can keep prices down once and if these therapies hit the market. Because if you don't have the competition, they're just going to set the price as high as they can, right? So I do hope that if these therapies turn out to pass through phase 3, that we will see more than one product on the market.
But I think it's fair to say, Agnete, that from your own work, the manufacturing of these cells is actually relatively quick and should be relatively cheap. So it's the manufacturing in theory shouldn't be expensive.
No, the manufacturing itself is not so expensive. The surgery, of course, is relatively expensive, but it's more or less the same as the DBS surgery. So there will be need some training on site, but DBS has been rolled out. So I do think therapy like this can also be rolled out to a similar extent.
Thank you, Agnete, and I'm sorry for being so tall and forcing you to look at a crooked angle. So we've had a good discussion today on cell transplantation, but Alan, if I come to you now, cell transplantation is not the only option that's being developed. And we're talking about competition between biotech companies here, but there's also competition between different approaches. So we have gene therapy approaches, and we've got adaptive DBS coming online as well. Can you speak to some of this, please?
Thank you. No. I'll speak up. Thank you very much. Oh, hello. Thank you very much, Simon. My name's Alan. I'm a neurologist. My interest in neuroregeneration is not cells, but GDNF, which is a growth factor, which is a bit like baby bio. You put it on and it sprouts your nerve endings. And the way we're currently trying to deliver that is by having again, you have to put it in surgically into the brain. And you're putting in a gene, and the gene gives off the GDNF. So it sits there expressing it and hopefully will restore dopamine neurons. And that's a contender against cell therapies. But the other contender that's surgical is deep brain stimulation. And that too is a very dynamic field. So these things are moving forward in tandem. And the benefits of deep brain stimulation is, you turn the box on and the patient's better. So they're struggling to walk, turn it on, and the person with Parkinson's is striding out of the clinic if it goes well, rather than having to wait 2 or 3 years. And it's a proven therapy. So one of the things that you're giving up if you go into these type of trials is in addition to the risk of surgery, you're delaying the possibility of benefit. So you're committing, therefore, to living with movement fluctuations for maybe 18 months while the trial goes on. And that's a really tough ask to say, do you want to put up with that? You might be in placebo, but these cell therapies are going to take 18 months, 2 years, 3 years to come through. So there are quite a lot of decisions. And now we've got adaptive DBS, which is the electrical signal in actually changes as you need it. So it's sort of personalized medicine. When you need a bit more electricity, you get it. When you don't need as much, it drops down. So they are going to be competing with each other, but they all do need surgery, and the surgical risks are not insignificant. You've got a 1 in 100 to 1 in 50 chance of having a hemorrhage. And there's also the risks of infection, introducing infection to the brain. So it has to really be thought about.
Indeed. And someone who's just recently thought about this is Dr. Jon Stamford. Jon is in a unique situation of having researched dopamine and Parkinson's his whole career, then developing the condition. He's not alone. There have been a few researchers with Parkinson's researchers who have gone on, such as the incidence of the condition. But Jon, you've also recently had DBS. Can you speak to that?
Certainly, yeah. Well, my experience, let me just put it in context. I've had Parkinson's or post-diagnosis for 18, 19 years now. So, yeah, fair old whack, really. And we discussed the option of DBS about 4.5 years ago with my children. And I asked them, what would happen in relation to risks, for instance, of DBS? What would happen if I died? And my youngest said, well, I'd be a lot richer. Which may or may not be true, but wasn't quite the parental backup that I'd actually been looking for. But the DBS itself has worked. In my case, I've been very pleased with the results. It's and I think a lot of it is down to expectation management. I think you have to make sure that you select the right patients. That it's not for all people by any means. In my case, said it was done in November the 2nd, if I remember rightly, 4 years ago, and at the hospital I was at, they turfed everybody else on the ward out at the weekend so they could go off to fireworks parties and things like that. They left me in on my own, and they kept bringing me magazines all about travel and things like this. And I was thinking, well, that's wonderful, but I can't go anywhere if you. Anyway, and so I fixed up a camera at the far end of the ward and connected it to my daughter's phone, and I walked up and down the ward. I said, "Watch this," post-op. And she said, and I heard her very clearly at the end of the thing of the room, and she was sobbing. And I said, "What have I done? Why are you upset?" And she said, "I've never seen you look so good, Dad." She's obviously never seen my Sinatra impression. But no, in my case, as I said, it's worked very well. I'm a bit of an old dinosaur. I don't have an adaptive DBS. It's just the old vanilla DBS, as it were, from Boston Scientific. And there are other companies, plenty of other companies who've had DBS. Simon, stop me at any minute, so I'm just going to ramble forever otherwise.
Well, just I'll let you continue rambling. What was your experience of the surgical procedure? And Andy has spoken about this afternoon, but how was yours?
Well, I was under for 9 hours, I'm told. My experience was very positive. I was aware of them starting to inject my arm with propofol, and the usual thing, you get to about 7, you count up to about 7 before you lose consciousness. And then you have no perception of time in anesthesia at all. If you go under general anesthetic, it's as though it's instant. So I woke up and several microseconds later, and obviously thought, it's gone wrong. They've obviously not done the procedure because why would I be awake so soon? So I reached up to my head and the nurse shouted, "Leave your scars alone." And it's a lady, and she was very forceful. But as I said, the procedure had gone ahead. Other than inability to remember the lockup code for my phone, it was all pretty good.
Very good. I'm going to open it up to the audience for some questions. Rory, in the middle of the
Thank
crowd
you.
there.
I'm just interested in timescales here and the dilemma it presents to cure Parkinson's as an organisation. Roger, all the scenario you painted of things going well and this getting approved, what's your estimate of how long that would take?
So if I've understood it correctly with the Blue Rock study, this will begin this summer. And they will recruit 102 patients, I think, to their trial. And so that will probably take about 18 months, I would think. And then I think the endpoint's 2 years. So that would be end of 26, 2028. So by the end of that year, so 2029, they would know whether it was a positive outcome. So if that was a positive outcome and they got a license for it, then it would then, assuming everything else passes through, then that's when it would become a therapy. That would become a therapy in America. I'm not familiar enough with how it would then translate across to others, but that would probably be the earliest point at which this therapy could get approval for use in Parkinson's.
They say a cure is always 5 years away, and that's 5 years away, so that's good.
Yeah.
And this is for you, Simon, or for Helen. Does that make what you fund a difficult choice? I mean, we seem to be still quite a long way from a pharmaceutical cure. do you begin to sort of shift your funds in certain directions?
Rory, I think that's a really important question. I think it's so important that all areas of research move forward in parallel, because, you know, Blue Rock is pushing ahead. Fantastic. We don't have to fund that. They are funding that. It's brilliant. Surgery is not necessarily the right solution for absolutely everybody. So having additional routes that could be mediating the disease, slowing the disease down or stopping it feels very important. And if that can be done through small molecules, that's really, really important. But the other thing I think that we're really aware of is that, you know, with the stem cell approaches that are being discussed today, that doesn't necessarily mean you're stopping the pathology of the disease, and that could still be active. So it's possible that you might need to be thinking about stem cell surgery plus a small molecule to work alongside each other. So really trying to understand all approaches is really important. But the critical thing is working on the biology of the disease. Simon, do you want to add to that?
Absolutely, yeah, 100% in agreement. Every year, Cure Parkinson's puts out a report in collaboration with the Michael J. Fox Foundation, looking at the entire pipeline for drug development and new therapies for Parkinson's, both symptomatic and disease modifying. And every year we consider cell transplantation in this format symptomatic. It's replacing lost dopamine as opposed to curing the condition. And
Is that fair? Is that, I mean, there's a big, you know.
well, that could be a question. That could be a question for the panel.
Yeah.
Roger, would you consider cell transplantation in this format disease modifying or symptomatic?
I would say it was both. So, you know, and you know, I made, I think the point this morning that, you know, in the pre-L-DOPA era, your life expectancy with Parkinson, I mean, I don't know because no one ever really looked at it, but your life expectancy was probably less than 10 years because you would actually be, was it? Yeah, because you know, because you just succumb to the condition because you had no symptomatic improvement. So if you take a good symptomatic therapy, it's not modifying the underlying pathology of the disease, but it is modifying the natural history of the disease. So, for example, most people are never cured of asthma, but I mean, you know, sadly, some people do die with brittle asthma, but most people live an entirely normal life with asthma because they have good symptomatic therapy. So I think that if the cell-based therapy works really well, then it will be fantastic for the dopamine aspects of it. Now, you know, we are starting to look at other problems with cell therapy. So Agnete is looking at, for example, some of the thinking problems in Parkinson's, whether you can go after that with the cell therapy around a different chemical. And so some of the other issues that, the other thing I just wanted to say was in terms of new innovations and combining therapies. So I was very excited that I've been approached by a company where we're about to start a trial, which is with a gene therapy, which is targeting the underlying problem in Parkinson's disease in some people, which is this GBA mutation. And this is going to be a gene therapy which you infuse into the bloodstream for an hour, and they've engineered it so it goes to the brain. So innovation is coming in this condition, and none of these are mutually exclusive. So, you know, whilst I've talked about this dopamine cell therapy and what it could do, if this gene therapy works as well, which is systemically given, you could combine these two. So I think disease modification, I think, is a term. I mean, the late Gerald Stern always used to talk about this disease modifying. Obviously, you know, we always talk about it in a positive way. I mean, you know, he would always say, well, it could modify you into a complete vegetable, and so, you know, it's not disease modifying. We always think of it in one direction or the other. But I do think good symptomatic control combined with something that may turn the dial and slow things down, and to all intents and purposes, that would do the job. So they're not mutually exclusive, I think, these treatments. And I think we should be trialing everything because we never know where the hit will come, where the win will come.
Alan, same question to you. Disease modifying or symptomatic?
Well, that's so same.
So for similar reasons to Roger, I would say disease modifying, but to provide a counter argument in the style of scholastic debate, I would say that symptomatic. And the reason I'd say that is because you're not protecting brain cells with giving cells in. You're simply replacing still something that's there. The great benefit is by giving cells that may replace lost cells to give off dopamine, you might then be providing dopamine in a different way. So the problem with pumps, if you have duodopa pump or the Lexicon pump or produodopa, which give continuous levels of dopamine into the system, which is why it improves fluctuations. The problem is with that is that that's not the way the body works with dopamine release. It doesn't provide a steady, constant level. When you need it, it has what's called a phasic response. So you need the cells give off dopamine when you need it, and because you want to move, and then it drops down again. So your cell therapies might work in a better way, and they also may well not have the side effects like impulse control disorders, for example. And so they may well give you better symptom control, but I would argue that you're not slowing down or protecting neurons because alpha-synuclein is still in the natural dopamine cells and they're dying off. So I think it's more an exciting symptom improving therapy that may overtake pumps or DBS. DBS can cause lots of balance problems and falls, so it's no free lunch. So maybe cell therapies are the best for free lunch if you can cope with the immune suppressants. So
That's a good answer, yes. And Agnete, I'm going to just take what Roger said here. Something that we're very keen on at Cure Parkinson's is this idea of combinatorial approaches now. As opposed to just testing single drugs, we need to be thinking about, it's a complex condition, so we need to be thinking about targeting it in different ways. Can you just comment briefly as to what Roger was talking about with regards to the new areas that you're looking at?
Yes. So, I mean, we are well aware that the dopamine neurons will mainly target motor symptoms. And I agree that in that sense, it should probably be characterized as a symptomatic treatment. And Simon actually sets the standard here because Simon writes the yearly review on all new clinical trials in Parkinson's disease. And it has been characterized in that review as a symptomatic treatment,
Not just me. Kevin McFarlane, Gary Rappolo.
It's fair. It's fair, right? I think it's a dopamine replacement, but maybe a more advanced and hopefully more elegant dopamine replacement, which is self-regulating. Yes, so we are aware it targets mainly motor symptoms. We are looking into, we do know in some individuals, those individuals developing cognitive issues, there is another type of neuron that is affected. These are the cholinergic neurons located in the basal forebrain. It is known that these are lost in certain subtypes of Parkinson's disease. We are generating those in the lab, also now testing them in rat models. That could be a potential add-on therapy when you're transplanting the dopamine neurons. We are also doing actually drug screening in the lab because I completely agree also what Helen said. The ideal therapy would be that you can both replace the dopamine neurons and then stop the disease at the same time, right? We don't have anything that can stop the disease right now. This, I think, is a huge need in the field. We are using the exact same dopamine neurons that we're putting into the brains of individuals like Andy here. We use them in the lab also to screen for new drugs that can stop the pathology, stop the cell death of the dopamine neurons. And I think this is as important as developing the cell therapy and other surgical approaches.
Very good. Thank you very much. Question from the audience.
I've got two questions, if that's all right. The first is interesting you raised the topic of mutual exclusivity. So I was diagnosed with Parkinson's in 2017, age 37, and I see before me 3 people proponents of team stem cells, team GDNF, team brain surgery, DBS adaptive. And I'm not naturally a gambler, so I'm trying to work out which horse to back. And are they mutually exclusive? So, for example, if I put myself forward with a stem cell or GDNF, heaven forfend, if it doesn't work, does that preclude me having DBS down the line? Where would you go with that?
So this is a question I often get asked, actually. So currently, when you're developing a new therapy and then people are going into trials, we don't take people into our trials for putting cells or genes into their brain who've had DBS just because it's an added complication and we don't know if there's any interaction. There probably is not. It's the other way around is perfectly reasonable. So we did 2 gene therapy trials with dopamine gene therapies, and those patients have gone on to have, some of those patients have gone on to have DBS, and there's a paper which I don't know if it's out yet or is certainly accepted, showing that DBS works just as well in people who've had a previous gene therapy. And some of the people who had fetal dopamine transplants in the past have gone on and had DBS as well. So having an experimental therapy wouldn't prevent you from having DBS. DBS would stop you from coming into a trial for one of these at the present time. Now, if it got a license, then that would probably change. So I would say they're not. You can certainly have these combined therapies. People have also looked at GDNF plus cell therapies. So Claire Parrish and Lachlan Thompson in Melbourne in particular have done this, trying to get the best of both. So if you put the cells in and you put GDNF, you not only regrow the intrinsic dopamine pathway, but you then encourage the transplanted cells also to grow better.
In
So-
rats, we should add.
Yeah, it is in rats, but it's the principle. And obviously, if you then took this, I mean, the reason why people don't do this as a sort of opening gambit on your trial is the regulation's hard enough with the gene therapy. It's hard enough with the cell therapy. If you combined it, it would be, I couldn't cope.
Alan, just same question to you before we go back for your second question, Claire. But you're one of the investigators on the Regenerate-2 study, which is the GDNF AAV gene therapy approach. If somebody partakes in that study and there is no effect, they can always fall back on DBS afterwards?
That's correct. So exactly as Roger said, if you've had DBS, you won't be able to go into a stem cell or GDNF study because it's too difficult to evaluate what's happening, what's helping what because of the confound. But certainly in our last GDNF study, where we then had to stop the GDNF because we were giving it intermittently via a port, then many of those patients have gone on to DBS since. So the fact you've operated on the brain, you can operate twice if you can stand it. But to answer your which camp to go with, so now this is true scholastic debate because I'm going to be a divided self and argue again for DBS, and then I'm going to argue for GDNF. So DBS, you get an immediate response, and it's an evaluated and proven therapy, and it's funded in the UK. So if you want to come and see me for DBS, we'll sort it out in 3 months, okay? So that's good. But downside, side effects. So balance problems are important. So we might help your stiffness and slowness and off periods, but we might have you fall more. And it can cause changes in verbal output, which is quite odd. So people worry about the speech problems, but it's not just the slurring of speech. It's also that you just become less verbal. It's rare, but the spouse might come back and say they just don't say anything anymore. So that's very disturbing. So that's the pros and cons. GDNF, no disrespect for the cell therapies, but GDNF, I would say, is restorative rather than just symptom improving if one argues that cell therapies are only symptom improving, because you're actually trying to bring back to health your own neurons. You're regenerating your own neurons, not someone else's that you've implanted in. So I think that is, of the two, that probably is the only one that fulfills the definition of a truly regenerative therapy. But I think in terms of which one to go for, I think either, because what you really want is good symptom improvement without side effects.
Question number 2.
So I really love Roger's the work with that long-standing epidemiology study looking at individuals, such that in the case of Andy's surgery, you've then got a really well-documented trajectory of where your disease was going, and then you can see the impact. I'm just wondering, are there any plans afoot within the UK to ensure that each and every one of people with Parkinson's, or at least a certain number, maybe geographically distributed across the country, are being more observed and more quantifiably observed, so that we've got ready cohorts to be able to pick people from maybe a line of PD frontline? I don't know.
There are a number of things that are going on. Yes, we have got genetic cohorts with PD frontline. That's starting to happen. That's great. But also individual centres will also have their own cohorts. But in parallel with that, and linked to the multi-arm, multi-stage trial platform that's coming this summer, there is an initiative called Join Parkinson's Research. And that is going to be a registry of patients, which is a start. Longer term, on hearing that that was coming, my immediate question to the investigators were, well, can we add in a patient-reported outcome into that so that at least you're starting to collect relevant information from people and they're contributing to a data set? I don't know if that's possible yet or what that would look like, because it's literally launching, currently. But I think people who are interested in research, join Parkinson's research, will have more than just EJS Act PD. It will also enroll people to PD Frontline. It will also enroll people to ASPRO PD. It will also hopefully enroll people to Regenerate PD. It will also, there are all these studies that it can do eventually. But it literally, we're on week 1, I think.
It's not just that I think you've got a patient-ready cohort. I mean, for these experimental therapies where you have very few numbers, it's incredibly informative. So for our TransEuro study, you have 5-year running before you intervene. And so you can see, so people are bored rigid by the tests we're doing. So there's no practice effect. All of this is gone. And then you see if there's points of inflection, and that can be incredibly helpful. So the individual is their own control in these studies, which I think gives great strength to it. It's just trying to, and I think if we set this up and then people are in it, it also, as Andy was saying, he's in the TransEuro observation. He doesn't have the transplant, but comes along and has one later on. So being in these studies and not refusing, but declining to be taken a therapy at one point doesn't stop you joining another. So I think they're very, very powerful.
And hugely important. And I think it comes back to the question I asked Andy earlier about the potential for that long-term follow-up as part of TransEuro. Because if you do take part in these studies, whether you're looking at Alan, whether you're looking at Roger, whichever route you're going for, committing to that long-term follow-up is absolutely vital, because the more we can learn, that data is helping us to change the future.
Before we take another question from the audience, are there any questions online?
Oh.
Or maybe we'll take Michael's question while we're waiting for that.
We're told that the only current disease-modifying treatment is exercise. Now, at the Kyoto WPC, Jun Takahashi, when he gave a speech about stem cells, said that there was going to be an exercise component to anybody who went through stem cell therapy. I don't know in practice whether that happened. Agnete, I'm interested, did you consider exercise as part of the protocol in the Lund study?
It's not something we've discussed, but it's not a bad idea actually, because it is true that exercise certainly in general is beneficial for motor symptoms, but also in animal models has been shown that it may enhance the integration of the graft and speed up the function of the graft. Luckily, I know that Andy is a very active ping pong player, participating even in competitions around the world. So I know that
And he
you are.
wins them as well.
Andy is doing the exercise part. June, maybe you can answer to maybe, did you incorporate any exercise in the Kyoto?
Patient. We haven't changed the rehabilitation program, but I do see, I agree. And rehabilitation and the therapy have the good effect.
Just for the folks that
Functional extension.
This is an animal model, so yeah, okay.
Expect the same thing will happen in the patient.
But have you encouraged your trial participants? No, but I mean, it's not a bad idea. I think we try to keep the trials relatively simple in the beginning, but I'll take your point. We should consider that.
I mean, I think the slight problem is in the preclinical, the animals, probably the animals which are active and the ones which are inactive is a bit artificial in human existence. We're all pretty active. We're probably more active than the inactive rats, even if we regard ourselves as rather sedentary. So I think it's difficult, and it's quite interesting in the world of spinal cord repair, where this is a big thing about trying to repair the spinal cord and get exercise. You actually have to tailor your rehab to the type of repair you're trying to get. So if you just give indiscriminate rehabilitation to people with spinal cord repair, or at least experimentally, it's worse than if you actually target it. So there's lots to be done here. I think it's just quite difficult to build it into a trial at this stage. And to be honest with you, the people who join our studies, they're not inactive people.
Andy, you raised your hand. Do you have a question or do you want to
Yeah.
promote ping pong? Or...
Well, promote any activity and exercise for two reasons. One, just to get through the surgical procedure of any kind. It's best to be as fit and healthy as you can. And I'm surprised, as I said, how hard it hit me for quite a while. But thankfully, it's a quick recovery once they reduce the medication. But more importantly, I was really curious, and I'd love to know what I could do personally to sort of nurture and encourage these to be as strong and healthy as they can be. And yeah, I'm still trying to find out, what's that other than exercise in general is good. But yeah, is there anything else specific, as Roger's highlighted there? I think there's some in stroke patients, apparently physiotherapy is kind of treatment that can help. So yeah, I'm really keen about that nurture aspect, if that's feasible or going to help in any way.
Agnete, preclinically, beyond the neurotrophic work that as Roger was talking about in Australia, what else are people looking at?
Well, I mean, exercise, if there's anything you can do is I think the only thing that has been shown you can do actively yourself. Other things have been tested, like you can strip the cells of certain surface markers, which makes them kind of enter more easily into the brain tissue. This is a genetic modification you make on the cells. This has not been tested in clinic yet, but maybe something we'll maybe be seeing in version 2 products, because it is an issue, as we've talked about several times, this very slow effect of the graft. If we can speed that up in some way, will make a big difference to the trial participants. So I do think this is some of the things we'll be seeing in version 2 products will be modifications of the cells that will speed up their maturation. And probably many of them will be genetic modifications. So you don't need to do anything in that case. It would be up to the cell manufacturers.
We'll take one last question from the audience and then we'll take some online questions before we finish up. Okay, 2 more questions.
Sorry. Thank you very much to everybody. I found it very interesting. I'm very interested in what tests you use to decide whether your treatment is effective or, for the individual and in the population, because as you very well know, and I only know just from experience, from knowing lots of people with Parkinson's, how very different the disease shows itself amongst different people, whether it's physical. The physical bit's easy to test, right? You don't shake so much, you don't kick. But what about the mental side, which for me, I think is just as important? And what sort of tests are you developing to see whether these therapies help with any sort of mental confusion or forgetfulness and all the other things that we know are symptoms of Parkinson's?
Yep, so preclinically, Agnete talked about this in her presentation, but maybe Roger and Alan can speak to this.
Yeah, I mean, it's a very good question, and it's a very moot point because there's what you can measure, what's useful to measure, and what the regulators will take as a reasonable measure. So these are not all the same, and it depends a bit what you're doing. If we take dopamine cell therapies, for example, obviously what we're trying to improve is the dopamine responsive elements of the condition. So, some cognitive deficits are due to dopamine abnormalities, not necessarily deficiencies, but high dopamine states in some parts of the brain. So what we would be targeting mainly with our dopamine cell therapies as an endpoint is motor measures, which is what are the most sensitive to it. So that's one way. The second is that often they are quite subjective based on what people assess when they see people. So they give people a score. So it's an objective number, if you like, but it's subjectively derived. You can then do, and Alan could probably talk to this about, electronic apps, wearables, and collect that data, but that's not really recognized by the regulators as being useful. And then obviously the final thing, which is the most useful, is how do you feel? So there's no point me saying, look, you're absolutely crackerjack, you're fantastic. I've looked at your dopamine scan, it's back to normal, your UPDRS has improved by 30 points. I don't really care that you say you feel absolutely lousy and it hasn't helped you at all. I'm telling you, you're much better. So then it becomes quality of life, which of course is very subjective. So it's very difficult. And if you're going to go after cognition, it might be easier because there are standard cognitive measures you can use in Parkinson's, which have been well validated. So it's not easy. And at the moment, I would say that the field has largely settled on what the regulators regard as the most important, which is things like the amount of time you spend on, the amount of time you're spending off, and then certain measures which we use. None of them are perfect. And at this stage, you know, there is a great debate as to whether we can improve on that. But it's also getting the regulators on board to accept, because ultimately the companies are going to want to have something which the FDA would approve. And if you say, well, I've got it improves on this, and they say, I don't care, that's not what we take as our licensing measure, you've got a problem. So Alan, I don't know if there's anything else you want to add.
But I mean, there is cognitive testing as part of the STEM PD trial, right? I mean, but they're all secondary measures.
So, so in terms of your question, which was how do you help these other non-motor aspects? You, as people have said, you can use rating scales. As important as cognition is mood. So if you're very depressed, then life's going to be awful, whatever. So you can do rating scales, but to flip it around, because you talked about exercise. So here's another thought. If you're depressed, your Parkinson's will be less well controlled, or if you're anxious, you won't move as well. So the best example I have of this is in the prior GDNF trial in about 2014. This older man came to be evaluated, and I thought he was too advanced. And he was pretty lonely. He shuffled into the clinic room. He looked a bit shabby. Anyway, about 2 years later, he came back to clinic to talk to me about DBS. And he strode in through the door. He looked very, very dapper. And I thought, how did I get it so wrong? I thought he was terrible. And what's gone on here? And then about 2 minutes after that, this rather attractive young lady called Elizabeth comes through the door and said, oh, may I introduce you to Elizabeth, my new wife? And it just showed the point. I mean, when the social situation was better and his happiness was better, his Parkinson's was better. And I have another patient who's had Parkinson's for 40 years, who I used to know when she'd had arguments with Bob, because they were pretty feisty. And then she'd ring in to say her Parkinson's, she was stuck on, couldn't get off the couch. And then when she was getting on better with Bob, she could move. So these other things are very important.
There's that famous case of Peter Baines, where you had somebody who was waiting for deep brain stimulation, and because they were having to wait so long, the family bought the person with Parkinson's a dog for Christmas to keep them company. And the consequence of that was she had to look after the dog and then take the dog out for a walk. And over 6 months, she, improved to such an extent she came off the DBS waiting list. So Peter Baines' conclusion was that everyone with Parkinson's should have a trial of a dog before we enter them for any advanced therapies.
I'll just add that Bob is not his real name. One last question from the audience.
Just, I was just wondering, because one thing we didn't discuss is the role of alpha-synuclein seeding and spread in cell transplantation or GDNF. Does that going to make any difference to these?
Yeah, so this is the idea that what goes wrong in Parkinson's is the protein which lies at the heart of an alpha-synuclein can spread from an affected cell into a non-affected cell and then template, if you like, the normal alpha-synuclein in that cell to become abnormal. So the disease spreads from cell to cell. So it's certainly fair to say in the fetal transplant work, so this is not stem cells, this is fetal dopamine cells, after 10 years after the transplant's been put in those patients who have died and they've looked at it pathologically, the transplant, some of the dopamine cells in the transplant have alpha-synuclein pathology. And in fact, that's where the whole protein spread theory began. And by 24 years, which I think is the longest surviving transplant, about 20% of the dopamine cells have alpha-synuclein in it. So on that rate of accumulation, probably 40 to 50 years after a transplant's gone in, it would have lost all of its dopamine cells. But by that stage, something else would have got the person with Parkinson's. So it is a potential risk. But the rate of accumulation is not sufficient, I think, to mitigate against using this treatment. And actually, one of the advantages of stem cells, as Agnete can speak to better than me, is you can obviously engineer them. It's one of the beauties of these things. You can change them. So as Agnete said, you can change them so they avoid the immune system. You could also knock out alpha-synuclein in these cells. And there may be reasons not to do that. They may affect their function. But if those cells you transplant do not have alpha-synuclein in it, there is no way they can acquire alpha-synuclein pathology because you've got nothing to template. So these are questions which people have asked. People have looked at it. I would think it's fair to say the alpha-synuclein knockout, when people have looked at them, behave pretty normally. So it might be something that's worth pursuing. For me, I don't think it's a big enough issue to do that, really. I don't think this will be a big issue. And I would suspect that with GDNF won't affect the accumulation of alpha-synuclein, and our cell therapy will not stop the accumulation of alpha-synuclein in the brain, either within the nigrostriatal pathway or anywhere else. It's not curative, it's simply repairing that bit of the brain.
And as you've pointed out many times before, Roger, what you could do is you have your cell therapy or your GDNF to get a really good symptom response, maybe better than DBS without the side effects, but you also take the pills as well to help with synuclein. So you take your monoclonal antibody as well. Yeah. You take your ambroxol to help your GBA system. So there is no reason why you couldn't combine it. And if you think of the HIV story, when I was at medical school in the mid-1980s, I mean, it was a horrific death for people over short periods in time. And for a long time now, it's been a condition that's effectively cured. I mean, you can live an entirely normal life expectancy with HIV, and that's happened because you take multiple different tablets, and so there's a synergy between those tablets, and you get your big effect. So I think in the future the answer is polypharmacy, multiple treatments.
So on that note, just finally, I'm going to finish with two questions from online. And the first question, straight back to you, Alan, is the combination of cell transplantation and DBS. The best of both worlds is what the questionnaire is asking. If the cells are going to take some time to mature, having DBS at the same time, is this a viable approach?
The answer is yes. And if I mean, I think first you'd have to do that in trial. And we were talking only yesterday, because we're looking at that mid-stage disease. And the reason we look at mid-stage is because if you want to see an improvement over baseline, you have to be bad enough to see that uplift. But what you could do to prove the cells work is you implant people with DBS. They have the benefit of getting better now. And then you wait for the cells to, when the point when you think they're going to kick in, say 18 months later, and then you just turn the DBS system off, and you don't tell the patient whether you're turning it off or not, so they're blinded to that, and you see whether their benefit's maintained because the cells have taken over the symptomatic effect. So I'd quite like to do a combined trial, but Roger refuses to work with me, so he's ready.
Well, we're about to add alcohol to this relationship, and we'll have drinks just after this last question. Roger, one question, quite astute question from the online crowd is: what is the risk of graft-induced dyskinesias in these new trials?
Well, we don't know, because you obviously we're in very early stages, and it depends what your theory is as to why people develop these in the first place. So just for those who are not aware of this, in the fetal transplant, so this is where people, as Agnete very eloquently said in a talk, this is where people receive transplants of the fetal midbrain. So this was the bit of the brain where the dopamine cells develop, and which are the ones which subsequently degenerate in Parkinson's. Now, obviously, that transplant contains the dopamine cells, but it contains lots of other cells, because the majority of the cells in that part of the brain are not dopaminergic. And so the theory is that some of the patients who had fetal dopamine transplants subsequently developed the dyskinesias which you see with L-DOPA, but in the absence of L-DOPA and the presence of a transplant. So they came off all their medication and they still had the dyskinesias. So at one point they were called runaway dyskinesias, which was a very unfortunate term because it implied that once they started, they just galloped away and you were left chasing these dyskinesias and the patient as a result. But actually they were just driven by the transplant, and it was thought that the reason for that was that one of the contaminants in the transplant which were not the dopamine cells, obviously they were the functional cells, was these 5-HT or serotonin nerve cells, which develop right next door to the dopamine cells. And these serotonin neurons don't have the same regulatory network. They can still take up some of the drugs, but they can still underlie the development of these dyskinesias. Now, with these new protocols that Agnete and other people have developed, there are no 5-HT neurons in these stem cell transplants. There are none. And we know that now because we have technologies which can look at each cell in it and we can't find any. So if that theory is correct, that those 5-HT neurons drove the graft-induced dyskinesias, then the risk of getting it with a stem cell transplant would be zero. And to date, in the trials that have taken place, there have been no graft-induced dyskinesias seen in any of the trials today, and that includes our own, although we're obviously at a much earlier stage. I don't know if Agnete, if there's anything more.
Yeah, I mean, in the two reports, 24 months, Blue Rock and Kyoto, they have reported no graft-induced dyskinesia. So I think that's very promising.
Wonderful. Okay. Well, I'd like to thank our panelists for a fantastic session. I'd like to thank the Cure Parkinson's team for organising today's event. And I'd like to thank you, the audience, for attending, those folks at home as well.